Okay, so welcome back to this next video in which we are discussing cholesterol metabolism. Okay, right. So we're in the process of discussing intracellular cholesterol homeostasis, how cells, both peripheral cells and hepatocytes, control the amount of cholesterol that they have. Okay, and we've just been discussing the SREBP pathway, okay, which is this pathway which responds to the cell having too little cholesterol. And we've seen that the overall effects of the SREBP pathway are that when cholesterol is too low, you increase the expression of these two important proteins, the HMG-CoA reductase enzyme, okay, and that's brilliant. We know exactly what that will do. That will catalyze that crucial step in the cholesterol biosynthesis pathway, which is rate limiting, and therefore if we have more of these enzymes, we're going to produce more cholesterol. Okay, so cholesterol biosynthesis is going to be increased in the cell now, and that's going to help to bring cholesterol level back up. Okay, the other thing that I've said is crucial that is going to have its expression raised by the SREBP pathway is this LDL receptor. Now that's a newbie, we haven't talked about the LDL receptor before, okay, and this is going to be involved in endocytosing LDL particles. So we've discussed LDL particles. They are lipoprotein particles within the blood that are particularly high in cholesterol. Okay, they have a lot of free cholesterol in the phospholipid monolayer, and they also have a lipid core that almost entirely consists of cholesterol esters. So they are these stores of cholesterol that are in the blood, basically, okay? And they are the main form of cholesterol within the blood, so other lipoproteins contain very little cholesterol compared to how much is contained within the LDL particles. Okay, and we call this store of cholesterol in the blood in the form of LDL particles the LDL pool. So basically, putting these LDL receptors on the surface of the cell is then going to allow the cell to endocytose LDL particles and harvest the cholesterol from those LDL particles, and hence use that cholesterol to raise cholesterol levels within the cell. Okay, and it's this pathway downstream of the LDL receptor, the pathway by which the LDL receptor leads to the endocytosis of LDL particles, that we now want to turn our attention to. Okay, so this pathway has a special name. It's called the LDL receptor mediated endocytosis pathway. Okay, so LDL receptor, because it's going to be done by the LDL receptor, okay, and it's going to be endocytosis of LDL particles. Okay, so LDL receptor mediated endocytosis. Right, now I can't really credibly talk about LDL receptor mediated endocytosis without giving you two important names, the names of the people who discovered this pathway, and these people are very, very famous. They also discovered the SREBP pathway, so they contributed a huge amount to biology. Okay, and they won the Nobel Prize in Medicine for their contributions. Okay, so the two very famous names associated with this pathway are Brown and Goldstein. Okay, and as I say, they also discovered the SREBP pathway, so they did a lot in the field of cholesterol metabolism. Okay, right, so it's good to know their names. People expect you to know their names. Right. Okay, so let's now talk about the LDL receptor mediated endocytosis pathway then. So I'm firstly going to start off by showing you the basic structure of the LDL receptor then. Okay, and I'll show you how it can bind to LDL particles by binding to the ApoB100 apolipoprotein in LDL particles. And then what we'll talk about is the endocytosis, okay, and we'll firstly talk about endocytosis in general, and then we'll talk about endocytosis of the LDL receptor. Okay, right, so let me firstly show you the LDL receptor. So the LDL receptor is in the plasma membrane, so now I'll draw the cell membrane here. Okay, so we'll say this is the extracellular fluid side of the cell membrane here. Okay, and we'll say that this is the cytoplasmic side of the cell membrane here. So this is the cytosol down here. Now, the LDL receptor, again, it's a single protein. It's not a multi-protein complex, which simplifies matters. It's just a single protein, so it will have an N-terminus and a C-terminus. And it only spans the membrane a single time, so the N-terminus ends up on the extracellular fluid side, and the C-terminus ends up on the cytoplasmic side. Okay, so I'll put the N-terminus here, and then the first portion I'll draw like so, and this is the portion that's going to be important in binding to the LDL particle, and that's why I've made it in that curved way, so that we can put an LDL particle in there in a moment. Okay, then there's another important domain that I'm going to put on here. Okay, and then you'll have the membrane spanning alpha helix and the C-terminal domain intracellularly here. Okay, so firstly let's talk about this ligand binding domain here. Okay, so the ligand binding domain is going to bind to the ApoB100, which remember is that massive great apolipoprotein that is the only apolipoprotein in LDL particles. And it basically consists of 7 repeat domains here. Okay, and these 7 repeat domains are known as the ligand binding repeats. Okay, so these are called the ligand binding repeats, and these 7 domains, which are all repeats of one another, okay, together they make up the full ligand binding domain. Okay, so there are 7 of them in total, and they're called R1 through R7. So the first one here at the N-terminus, that'll be R1, then R2, then R3, then R4, then R5, R6, and R7. Okay, right. So those are the ligand binding repeats, which together make the ligand binding domain, which will bind to the ApoB100 of an LDL particle. Okay, right. Now just before I put the LDL particle attaching on, I'll just show you this other domain that I've highlighted here. Okay, so I'll colour it in orange, and this domain here is called the beta propeller domain. Okay, now for our purposes it doesn't really matter that it is actually a beta propeller, but I'll just explain what a beta propeller actually is. So basically, a beta propeller domain, they're found in lots of different proteins, and basically the reason they're called that is that they look like propellers, so the sort of thing you might have on the back of a ship. Okay, and basically the blades of the propeller in a beta propeller domain are made up by beta strands all in parallel making little sheets. Okay, so that's why it's called a beta propeller domain, because it looks like a propeller and the blades are made by beta sheets. Okay, for our purposes it doesn't matter that it is actually a beta propeller. Okay, we will see that what this is important in doing is it's important in releasing the LDL once the LDL receptor LDL complex has reached the early endosome, but I'm now getting far too ahead, so we'll come back to the beta propeller domain later on, but for now just know that it's there. Okay, right, so let's talk firstly about how LDL particles can bind to the ligand binding domain of our LDL receptor, and this full thing, I'll just label it up, it's the LDL receptor, which often people abbreviate down to LDLR. Okay, now let's put an LDL particle onto our LDL receptor then. Let's bind an LDL particle onto our LDL receptor. Okay, so specifically it's the apolipoprotein of the LDL particle which binds to the ligand binding domain of the LDL receptor. Okay, so remember LDL particles only have that giant single apolipoprotein in their phospholipid monolayer, okay, which is the apolipoprotein B100, which I'll colour in turquoise here. Okay, so here's our LDL particle, and that portion on the surface of the LDL particle, this is representing the apolipoprotein B100, which is going to bind to the ligand binding domain of our LDL receptor. Okay, right, so that's how an that's how an LDL particle can bind to the LDL receptor then on the surface of the cell. Okay, what we now want to ask is how can this be endocytosed? Okay, right. Well, first I'm going to tell you about endocytosis. Okay, now endocytosis is a pretty controversial topic, okay, because what we used to think is that endocytosis was cargo-driven. Okay, now what does that actually mean? Okay, well, the LDL receptor with the LDL bound is an example of what we would call cargo for endocytosis. It's something that can actually be endocytosed. And our old view was that endocytosis would occur in portions of membrane where there was cargo present. Okay, however, that view is kind of being phased out in favour of there being certain portions of the cell membrane which are endocytosed all the time anyway, okay, whether or not there's actually cargo present there. And then the way that you endocytose the cargo is that the cargo, such as the LDL receptor with the LDL bound, will end up going into those portions of membrane. It ends up sticking in those portions of membrane that are being endocytosed, and then it will end up being endocytosed almost by accident, basically. And that's the view that we are moving towards, okay? That's the more now generally accepted view. But for a long time it was thought that endocytosis was cargo-driven, that it would occur at a portion of membrane where the cargo was, whereas now it seems as though instead there are portions of the membrane that are being endocytosed all the time, okay, regardless of whether there's cargo there or not, and then all that you have to do to get cargo endocytosed is just locate the cargo in those special portions of membrane. Okay, so that's what I'm going to tell you about with endocytosis. Okay, so let me draw this special piece of membrane that's going to be endocytosed then, and I'll colour it in yellow. Okay, now I'm going to go from representing the phospholipid bilayer as two lines in parallel to just representing it as one line. Okay, so this now represents the phospholipid bilayer. Okay, I'll just make the picture easier to draw. Okay, right, so let's say this portion in yellow is one of these little patches of cell membrane that is being endocytosed, that is going to be endocytosed. Okay, so what's thought to happen is that a complex of proteins known as the adaptor protein complex 2 binds on the underside of this membrane, basically. Okay, and what it actually binds to is still a little bit controversial, but it binds to something there. Okay, so adaptor protein, whoops, I've missed out the complex. Okay, adaptor protein complex 2 it should be. Okay, so move that 2 down here. Okay, and for short, the adaptor protein complex 2 is just abbreviated down to AP2. That's how I managed to miss out the complex. Okay, right, so we'll denote the adaptor protein complex 2 then in purple here. Okay, so I'll just represent the adaptor protein complex 2 as these rectangles that are binding at the underside of this special portion of cell membrane here. Okay, like so. So here they are in purple, and they are not an individual protein, they are a complex of protein, and they're binding to something about this special portion of membrane. Okay, so this special portion of membrane that is destined to be endocytosed must have something that allows the adaptor protein complex 2s to bind there. Okay, but as I said, it's still controversial what that is. Okay, right, so now what's going to happen is clathrin proteins are going to be recruited by binding to the adaptor protein complex 2s. Okay, now clathrin proteins can polymerize together. So when you attach clathrin proteins onto the adaptor protein complex 2s, they end up polymerizing together. However, so I'll initially show them unbound to one another. Okay, so at the moment they're not bound to each other. However, what can happen is these clathrin proteins can join with one another. Okay, so here are the clathrin proteins in green here. Okay, so they are representing clathrin. And basically, clathrin proteins can polymerize together. So these neighboring clathrins can now polymerize together. But when they polymerize together, they don't just form a long sheet like that. When they polymerize together, they form a great curved structure. Okay, so let me show you what this is going to do to the membrane. So basically, it's going to do this to the membrane. It's going to form an invagination like so. Okay, so once again, here are the adaptor protein complex 2s, and I suppose the first thing I should probably mark on is this special portion of plasma membrane here. Okay, then we have the adaptor protein complex 2s here in purple. Okay, and now these clathrin proteins have bound together basically but once they've bound together, they no longer make a flat structure. Instead, they make this curved structure like so. Okay, so here, these are the clathrin proteins. Okay, right. And you can see that it's now going to invaginate the membrane down like so, and this is going to create us what's known as a clathrin-coated pit. I'm not sure I'm going to actually, hopefully I'll be able to fit this in here. Clathrin-coated pit. Okay, now what's going to happen is this is going to extend basically. At the moment I've only got, 3 AP2s there and 3 clathrin proteins, but it extends. So imagine that this portion of cell membrane is bigger now. Okay, so zoom out a little bit and we've got more of the cell membrane that's going to be endocytosed. So now more adaptor protein complex 2s are going to bind here, and you're going to start getting those being added onto the polymer basically. Okay, so let me show you how this will now develop further. Okay, so it will now go into a much bigger invagination like so. Okay, and again I'll colour this membrane that's being endocytosed here in yellow. Okay, and it will have these adaptor protein complex 2s bound to it. There's 1, there's 2, there's 3, there's 4, and there's 5. And I'll colour these in vivid purple here. Okay, and each of the adaptor protein complex 2s will have a clathrin protein attached to it, and they've all polymerized round together. And as you can see, when they polymerize together, they're making this round shape like so. Okay, and the membrane is just being bent as the clathrin structure assembles basically. Okay, so all of these are separate clathrin proteins polymerized together into this clathrin polymer, and I'll colour them in green here. Okay, so this structure that we've now constructed, this more extreme clathrin-coated pit, if you like, it's now called a clathrin-coated bud. Okay, so we'll call this a clathrin-coated bud. So the clathrin-coated is obvious because it's coated in clathrin. So this is now a clathrin-coated bud. So we progress from a clathrin-coated pit here to a clathrin-coated bud. And note at the moment there's no cargo in sight at the moment. It's believed that this occurs whether there's cargo in here or not. Okay, we'll add in the cargo later on. Okay, but it's thought the cargo actually being in here is just almost an accident basically. Okay, so the actual endocytosis of cargo just occurs because the cargo ends up diffusing into here but then gets stuck in here when it ends up there, and we'll discuss that later on in a moment. Okay, right. So that's the clathrin-coated bud. Now let's talk about how you actually pinch this bud off to form a clathrin-coated vesicle. Okay, you might just think, well, isn't the clathrin just going to complete the job and form a nice sphere here? But it turns out that clathrin polymerizing in this way doesn't seem to actually have enough force behind it to actually pinch the membrane off. It doesn't have enough power, basically, to actually finish the job and pinch off the membrane. Okay, so instead it seems that another protein is really important here, okay, called dynamin. Okay, so let me just tell you a little bit about dynamin. So dynamin is a protein which forms dimers, and these dimers of dynamin proteins end up in a banana shape like so. Okay, so this is my picture of a dynamin dimer, like so. So this is the 2 dynamin proteins dimerized together. Okay, so here's one of the dynamin proteins, and here is a second dynamin protein. Okay, and both of these dynamin proteins have a special domain here, okay, which I'll colour in orange. So here is one of the dynamin proteins' domain, special domain, and here is the other dynamin protein's special domain. Okay, and these domains are known as the GTPase domains, okay? So these domains are capable of hydrolyzing GTP down to GDP and inorganic phosphate. So they're capable of catalyzing this reaction, GTP being broken down to GDP, and also an inorganic phosphate. Okay, now that reaction is very similar to the hydrolysis of ATP, okay? It's GTP rather than ATP, but it's similar, okay? It releases energy. Now, what have I just said? we need energy in order to break this membrane here, in order to pinch the vesicle off, pinch the bud off to form a vesicle. Okay, we need energy, and these dynamin dimers are actually going to provide that energy. Okay, so the first thing that needs to happen is these dynamin dimers actually need to bind to the membrane. Now, how do they bind to the membrane? Well, basically on their concave aspect here, okay, the protein has a lot of positive charge, okay, like so. Now, this allows it to bind to membrane because, remember, membrane, which is constructed out of a phospholipid bilayer, the heads of phospholipids have that negative charge. Remember, the phosphate group contains the oxygen with a negative charge, okay? So the phospholipid heads are negatively charged. So having that positive charge there allows it to associate with the membrane. And what's actually going to end up happening is dynamin dimers can assemble around this stalk portion of the bud here. Okay, so I'll just highlight this portion up. So this special portion here, this is what's known as the stalk of the bud, okay? And basically dynamin dimers are going to assemble around that. So if I now just draw this out, here is this stalk of the bud basically, like so. And now what's going to end up happening is because this stalk, the membrane is curved on this stalk, the concave aspect of the dynamin dimer is complementary to that curve. So what can end up happening is the dynamin dimer can attach to this stalk like so. Okay, so I'll just colour this in blue. This is one of these dynamin dimers assembled on the stalk. And then what ends up happening is they form helices around the stalk basically. Okay, so here's another one which will assemble on top of the other one, on the top of the first one. Okay, and these can go round in spirals basically around the stalk. So if you imagine the stalk being a little bit longer than I've just shown it, okay, this thing could continue on in a great spiral that will go round and round the stalk. Okay, and it's believed that what then happens is the dynamin dimers use their GTPase domains to hydrolyze GTP down to GDP and inorganic phosphate, and then the spirals can sort of contract around the stalk. So if I now show this on another picture, okay, so I'll draw the stalk a little bit longer so that I can exaggerate this point. Okay, so here is a dynamin dimer, and they'll form these spirals going round the stalk. Okay, so it will go round, and it might come back out on this side here. Okay, and it will continue going round, basically, like so. So these are all dynamin dimers. I'll just colour them in blue. And it's thought that once you have assembled a spiral like this of dynamin dimers, then all of the dynamin dimers can start hydrolyzing the GTP, releasing energy. And the thought is that this spiral can sort of contract and constrict around the stalk to pull it narrower, basically. So this can go to something that looks more like this. Okay, a much narrower stalk with much steeper dynamin dimers going all the way around, like so. Okay. Okay. So that's requiring the hydrolysis of GTP then to constrict down on this stalk of the bud, basically. Okay, right. And that's believed to be what then ends up pinching the bud off from the plasma membrane. Okay, and what you end up producing then is now a vesicle that's actually inside the cytoplasm, and I'll just move this up, which is still coated by the adaptor protein complex 2s, which I'll show here. Okay, so these are still the adaptor protein complex 2s, and then attached to the adaptor protein complex 2s, you'll then have the clathrin proteins. Okay, and it's important to understand that although we are just seeing a two-dimensional picture here, this in reality would be a three-dimensional structure. Okay, so the vesicle would in fact be a sphere, and the clathrin polymerized all over the place would be a sphere as well. Okay, so here are the clathrin proteins, and I'll just colour this in. So we'll have the adaptor protein complex 2s here in vivid purple. Okay, like so. And then around the outside, what you'll then have is the clathrin proteins, which I'll have coloured in green here. Okay, so this structure that we've now successfully pinched off from the plasma membrane is called a clathrin-coated endocytic vesicle. Okay, now it's very short-lived. Very quickly, what's going to happen after endocytosis has occurred is the clathrin coat is going to be removed. Okay, so this is a clathrin-coated endocytic vesicle. Okay, so very quickly after you have had this endocytosis process occur, and I should just say, this process of taking in an endocytic vesicle like this is what's called endocytosis. This pathway that we've just looked at, this is endocytosis, producing an endocytic vesicle from the plasma membrane. Okay, that's what's called endocytosis. So very quickly after endocytosis has occurred and you've got this clathrin-coated endocytic vesicle here, what will happen is the coat will be removed. So the AP2s will be removed from the membrane. Okay, and then the clathrin will be removed with the AP2s. Okay, so what you'll end up with then is just an uncoated endocytic vesicle. Okay, so here now, this is the uncoated endocytic vesicle. Okay, and I suppose I should colour in the membrane yellow because this is still that special portion of membrane that had something special about it that meant that it was destined to be endocytosed in this way. Okay, so as I say, this process is believed to occur whether there is cargo actually in this special portion of membrane or not. Okay, so it doesn't require there to be cargo there, it occurs anyway. Okay, however, what we now want to talk about is how then does the LDL receptor actually get involved in this process, because we want to endocytose the LDL receptor with LDL bound and have it inside this endocytic vesicle. So how does that occur? Well, let's go back to my picture of the LDL receptor and add something additional onto here. Okay, so I now want to add an additional domain that I didn't show previously onto the C-terminal side of my LDL receptor here. Okay, so I'll colour this special domain here in blue. Okay, and this special domain that's represented here in blue, this is what's called the sorting motif of the LDL receptor. Okay, and this is the motif that's going to help it to be endocytosed. Okay, now, this sorting motif is a sequence of 4 amino acids. Okay, and for short, this sequence of 4 amino acids is denoted the NPXY motif. Okay, and to understand this, you need to be able to read single letter amino acid code. Okay, so in single letter amino acid code, the letter N stands for the amino acid asparagine, okay, which would have 3-letter symbol ASN. The single letter P stands for the amino acid proline. Okay, X doesn't stand for anything, that just means it doesn't stand for any specific amino acid, it just means it can be anything in that position. And then Y is the single letter code for tyrosine. Okay, so this is just a sequence of 4 amino acids that is present on the intracellular side of the LDL receptor, which is extremely important if the LDL receptor is to be endocytosed. Okay, so if you have problems with this sorting motif, if you have a mutation in the sorting motif, the LDL receptor doesn't get endocytosed, and that will become important later on when we want to discuss familial hypercholesterolemia. Okay, right, so the X is here, okay, and that Y residue is a tyrosine residue. Okay, so basically this sorting motif is on the intracellular side of the LDL receptor, and it is this sequence of 4 amino acids which will be nicely visible in the cytoplasm, which is NPXY, asparagine, proline, anything, tyrosine. Excuse me. Okay, right. Now, what does this sorting motif then do? Well, basically, it allows the LDL receptor to bind to adaptoprotein complex 2. Okay, so let me explain how this is going to work. So we have put these LDL receptors on the surface of the cell. They're on the cell membrane. Okay, and they are free to diffuse about in the cell membrane, so they're not static, they can move around in the cell membrane. Okay, so they might just happen to diffuse into this portion of plasma membrane which is getting AP2 complexes binding to it. Okay, they might just happen to diffuse into this clathrin-coated pit here, okay, which has got the AP2 complexes and the clathrin coat beginning to assemble. Okay, and basically, if they just happen to diffuse into this portion of membrane, the sorting motif, this MPXY motif, can then bind to the adaptor protein complex 2. So let me show this nice and simply. Okay, so here is an LDL receptor represented here, and I'll colour it in red, and it has now bound to that adaptor protein complex 2 that is part of this clathrin-coated pit, and then it will get stuck there. Okay, so now it can't move. Now I want to stress, and indeed I've shown it here, that that LDL receptor doesn't need to necessarily be bound to LDL. This can occur whether it's bound to LDL or not. But of course some of them might just end up having LDL bound to them. Okay, and those are the ones we're really interested in. We don't care about the endocytosis of the empty ones. The ones we're interested in are the ones that actually do have LDL bound, okay? So this is the LDL particle shown here. And I apologise once again for the tiny size of this. Okay, right. So LDL receptors with LDL bound might just happen to end up in these clathrin-coated pits, and then they'll be able to bind to the AP2 protein, and that keeps them there. They're now stuck there. They're not going anywhere. And this means they're going to end up in the clathrin-coated bud. So if I show it again now in the clathrin-coated bud, here it is in the clathrin-coated bud. Once again, I'll colour in the LDL receptor here in red. Okay. Here is the apolipoprotein B100 in turquoise. Okay, and then finally they will then end up in the endocytic vesicle here. So firstly in the clathrin-coated endocytic vesicle, like so. So here's the LDL receptor in red, and the apolipoprotein B100 in turquoise, and then finally they'll end up in the uncoated endocytic vesicle as well here. So here is our LDL receptor with our LDL bound to it, like so, via the apolipoprotein B100. Okay, so basically my key point here is that the endocytosis of these LDL receptor LDL complexes is almost by accident. Endocytosis is occurring all the time in cells, okay, at certain special portions of membrane, which are these portions that I've been representing in yellow, okay, and it occurs whether the cargo's there or not. If you want to endocytose cargo, really the cargo just has to stumble across this site, basically, this site which is being endocytosed, and then keyly, once it stumbles on that site, it can't then get out, okay, because it has this sorting motif, this MPXY motif, which is a very common sorting motif seen in other proteins besides just the LDL receptor, which is capable of binding to the adaptor protein complex 2, okay, and therefore the LDL receptor with the LDL bound potentially ends up stuck there, and then it will end up endocytosed in the endocytic vesicle here. Okay, right, so we have now endocytosed an endocytic vesicle which has an LDL receptor with an LDL particle bound. Okay, we now want to see what's going to happen to that endocytic vesicle with the LDL receptor and the LDL particle in it. Okay, but we'll have a look at that pathway in the next video.